11, How Genes Are Controlled. In the previous chapter, chapter 10, we learned how DNA, based upon the base pairs of A, T, G, and C, we can determine what type of amino acids and what type of proteins we want to make. So basically, our 46 chromosomes have so many of our genes, and each gene is controlling how a protein gets made and helps determine our traits and our characteristics. So people's green eyes, blonde hair, tall, short, all that's determined based upon our genes and the proteins that they make. So the DNA determines the RNA determines the protein that gets made. So we are all different because of the different types of proteins, and therefore the DNA decides all this. So chapter 11 is going to be on how we're actually controlling it. And that's why the title is sometimes called Why Gene Regulation Matters. So we'll start with chapter 11. Now the first slide that I care for is this one. Now on this slide I only care for this guy right here called P53. Now that's a very, very special gene in our overall organization. The P53 gene is sometimes called the tumor suppressor gene. So P53 is sometimes called the tumor suppressor gene. Now here's what it really means. It means that if you ever mess up the P53 gene, you are now prone to getting certain kinds of cancers. So in essence, you really can't get a cancer of certain types unless what is damaged first, the P53 gene. That is your protection. It's the tumor suppressor gene. So as long as P53 is your night watchman and it's on the job protecting the rest of your genes and the rest of your chromosomes, then everything's fine. The moment that you have this night watchman sleeping on the job, P53 is not working right, you're now prone to getting cancers. Okay, next slide. Okay. Now, this slide says, how can a mutation in a gene lead to cancer? Well, it turns out that many cancer-associated genes are basically genes being turned off or turned on. Okay. Now, when you turn a certain gene on, then certain proteins are made. If you turn certain genes off, then certain genes and certain proteins are not made. So this is all about turning on genes and turning off genes. Let's take a good example before we move forward. When you have your grandma and you're pulling on your grandma's skin gently, and notice it's almost like not as elastic as it used to be. Matter of fact, when you pinch it, it sort of stays wrinkled for a while. That's because the protein in the skin, like collagen, for example, well, as you age, you tend to make less of this protein. Well, why are you making less of this collagen protein as you age? Because the genes have now turned off. So Nimish, are you basically saying that aging is all about certain genes being turned on and certain genes being turned off? And that's exactly what I'm saying. So aging is all about certain genes being on, certain genes being off, and when the ones that are on all of a sudden are off, or the ones that are off are somehow turned on, then we have issues. And that's what this slide's talking about. So having a mutation, and if you recall from a previous chapter, a mutation is simply a permanent change in a gene. Okay, so that's a permanent change in a gene from a previous chapter. Okay, next slide. Don't care. Okay, this slide, I do care. I care for that term in boldface called gene regulation. So gene control or gene regulation is about turning certain genes on and turning certain genes off. And that's the definition I want you to be aware of. That gene regulation is about just turning on or turning off certain genes. And by doing so, you're actually changing the function of how that cell actually works. Okay, moving on. Okay, this is another word down here at the bottom. It says gene expression. Well, that's exactly what we've learned in the previous chapter. Gene expression was all about taking DNA, turning it into RNA, and then turning it into some kind of protein. So gene expression is actually part of gene regulation. So when you're controlling a gene, you're actually controlling the expression of that gene in terms of, hey, do these proteins work now? Or no, these proteins don't work anymore. Okay, don't care. Now this slide is very important. This slide is page 198. Now why this slide's helpful to us is because it compares three cells: pancreas cell, white blood cell, and nerve cell. Now as you know, all of them have the 46 chromosomes. They all have the same 46 chromosomes. So whether it's a pancreas, or whether it's a white blood cell, or whether it's a nerve, they all have the same 46 chromosomes. But you already know that the pancreas cell is not like the white blood cell. It's not like the nerve cell. They're uniquely different. Yet they all have the same 46 chromosomes. So what this chart is saying is that it's based upon their function. So their function determines what genes are on and which ones are not. So let's take an example from the slide here, page 198. Here we see enzymes. Well, glycolysis, as you saw in a previous chapter, glycolysis has to do with metabolism and things like that. Well, look, all of them deal with that. So they all have a gene dealing with metabolism. Nerve cells have a metabolism, white blood cells have a metabolism, pancreas cells have a metabolism. So they all have that particular gene. Now what about antibody? Well, that's part of the immune system, right? Antibody is immune system. Well, only the white blood cell would have a gene because it functions in the immune system. Insulin. Well, insulin is from the pancreas. So there is a insulin gene that is working in the pancreas cell. But it's not working in others because insulin doesn't come from a white blood cell. It doesn't come from a nerve cell. Okay, hemoglobin, which is the red blood cell. Well, these are not red blood cells. So therefore, there is no gene associated with how a red blood cell would function. So every cell of our body's got the same 46 chromosomes, basically the same genes, but in some cells, certain genes are on and they're expressed, and certain genes are off. That's the story. Okay. I'm now on page 199, and that's with this picture that I'm moving to. Page 199. Now this is called an operon, and this was first discovered in bacteria dealing with lactose, the sugar. So the sugar lactose, which is found in milk, so the milk sugar called lactose, and this is how lactose is operating on the gene. Now this was discovered in a bacteria. So what we're going to do is we're going to look at the top picture and then we're going to look at the bottom picture. Now genes are not always on. Some are not. But usually a gene gets turned on based upon the need. So let's take an example of this top picture here. In the top picture on page 199, we see this gene right here. It's called a regulatory gene, and it's for the lactose sugar. So the lactose sugar. And here I see this red thing that actually is doing what? Blocking this part right here. So this gene is not turned on because lactose is not present. So if the sugar lactose is not present, then why would I actually use that enzyme? There's no reason to make an enzyme unless the sugar is present. So this operon is turned off. This gene is basically turned off because it's currently blocking the use of that part of the gene. However, let's say the sugar is present. So let's say lactose, the sugar, is available. Well, on the bottom picture, we see the little golf balls here at the bottom. The golf balls are the sugar, the lactose. And look what it does. It prevents this red blocking thing from kicking in. So this red blocking thing called a repressor does not block this transcription. And if you recall, transcription from chapter 10, the DNA chapter, was converting DNA into RNA. So in this case, the DNA is allowed to get converted to mRNA, which then, of course, will make the lactose enzyme. And of course, enzymes belong to the protein category mentioned long ago. So here in the bottom picture, we are going to make the necessary enzyme because lactose is present. So this is an example studied in a bacteria long ago that tells you how the operon or the gene is turned on or off. So this is called the lac operon, and it's named after lac for lactose. And that's how the top picture and the bottom picture read, page 199. Okay, all right, don't care. Now these two terms, I only care for the first one, and that's the promoter. Now don't worry about writing the definition, but I do want you to see the picture. And that was the picture I showed you a moment ago. And the green part here is the promoter. That's the starting point. So that's why it promotes. It starts off the process. You got to have somewhere to latch on to, and it knows to latch on after the promoter. Basically a starting point. Now how do you think that starting point, that green promoter, is going to be noted? It's going to be noted by a sequence. So if it's a TA, 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 where you have thymine, adenine, thymine, adenine, thymine, adenine. If you keep having that, sometimes they call that a TATA box because it's a TA, TA, TA. So there might be some kind of promoter here that tells me this is where I would start if I really needed to start. So that's what I care for in terms of a promoter. It's the starting point. I don't care for the rest. Okay. Don't care. And we've already talked about the red, which was the repressor a moment ago. So we've already discussed that. Okay, moving on. Okay, I don't care about this. Okay, this video is worthwhile watching. It is based upon page 201. So this video is worthwhile watching, but I'm going to be on this picture instead. Now this is page 201, and notice I have two terms here. I have this little bending part here, and it's going to be shown again in a little while. So we have enhancers that make it more likely for transcription to occur. And that's what enhancers are. They're more likely to make the transcription occur. Okay. All right, next slide. Okay, now in the last chapter, in chapter 10 DNA, we talked about DNA being changed over to RNA. So DNA turned into RNA. Now that was found in the nucleus. And remember, it was called transcription. Well, at that time I mentioned something called exons and introns. So exons and introns. Now introns are actually the garbage that we remove. So introns are waste. We get rid of it. And then all the exons are spliced back together. And at that time I called it RNA splicing in chapter 10. Well, that same concept is brought back here. So this is called RNA splicing. Now this is going to go with page 202's picture, which is the next slide. And that's this one. So again, as with chapter 10, we see our exons, and then we have this non-numbered area. These non-numbered areas are introns, and we're going to remove them. So the non-numbered items will be removed. But notice, this chapter mentions something different. It says that I have an option. Yes, I still have my RNA, my mRNA here, but I can actually make an option in terms of how I put it back together. That was not mentioned in chapter 10's video because they didn't talk about that. But in chapter 11, they're saying we're going to add some more variety, more flexibility, more diversity. And so therefore I now have 1, 2, 3, and 5, or 1, 2, 4, and 5. So I have some options of how my protein will look. So there's more possibilities of the outcome of that protein. It's like having more decks of cards, which means more possibilities for a certain card to show up. Okay, next slide. Next slide, next slide. Don't care. Okay, don't care for those. Okay, on this slide, I do care. This is based on page 203. So page 203. Make sure you watch this video on cell signaling. So page 203, watch the video on cell signaling. Okay. Okay, this one I do care for. This slide talks about cyclic AMP. Now, cyclic AMP is for chemicals that can't come into the cell, which tend to be water-soluble chemicals. So water-soluble hormones, water-soluble chemicals can't get in, and therefore they're going to use what's called a second messenger. And that second messenger is called cyclic AMP. So this video is worthwhile watching. So cyclic AMP. Okay, so cyclic AMP. Okay, next slide. Okay, make sure you draw a line. This is a new topic, a new thought. These are called homeotic genes. Now, these are the genes that tell where body parts should go. Why do we have ears not on the front of our head, but on the side of our head? Why do we not have ears in the front, but rather on the side? Why are our eyes exactly where they are? That's because of the homeotic genes. So on the next picture, page 203, you will see the homeotic gene for a normal fruit fly, where the eyes are here and the antenna is here. So eyes and antenna right where they belong. When you have a change in the homeotic gene, a mutation in the homeotic gene, you end up having extra pairs of legs where the antenna is supposed to be. Okay, that tends to happen. Okay, so that's a homeotic gene. Now, on your PowerPoint slides, there's actually a video to watch. So when you are watching the actual PowerPoints, in the PowerPoint, they have this video mentioning how homeotic genes determine whether the front of the fruit fly will become the front and why the back of the fruit fly will end up being the back of the fruit fly. So this is part of your PowerPoints, definitely worthwhile watching. Okay. Don't care. Don't care. Don't care. All right, this one's very important on a test. Cloning. There are two purposes for cloning. Okay, or gene regulation. Okay, so gene regulation affects two important processes. One is cloning and one is cancer. So one is cloning, one is cancer. Okay, this slide is important. So two regulations affect two important processes: cloning and cancer. Okay, next slide. Here's an example of a clone. Now in this example, which is on the bottom of page 205, this plant that they got a little piece from and then took a cell out of it and then grew it as an adult plant. So they took a seed, or a cell rather, a cell from this part and then grew a whole new plant. Now, these are not different plants. They're not the same type of plant. It is the exact same plant. Okay? So it's not the same type of animal. It's not a similar example of an item. It's the exact same. So this is a clone. And so when people ask me for certain plants in my house, I'll give them a piece. They can grow that piece, and then it turns into a plant. My plant and their plant is exactly the same genetically. Okay, next. Now this bottom part is of value to us. So the bottom part of this slide says seedless plants such as grapes, watermelons, oranges. Okay, they're all because they are made genetically. So what we're finding over time is that because certain plants are not so well off, like in Florida, if we get a freeze, or when the hurricanes come over and bring salt water into fresh water and it kills a lot of the plants, well, we have to have a plant designed a certain way where it's resistant to diseases. And that's what we're doing now. So much of our agriculture now is actually done through cloning strong plants to make more resistant to disease plants. Okay, so this slide is of value. Okay, you don't care? Don't care. Okay, this picture is very important. This is page 206. 206. Now this picture tells you 2 reasons for cloning. So on a test, what are the 2 reasons for cloning? One reason for cloning is reproduction, where you're actually making an exact copy of that animal. So that's a clone. So that's reproduction. The second reason for cloning is therapeutic. So if someone has a certain kind of cancer and you want to make sure that they're treated, well, if you have healthy cells from a stem cell, like from the umbilical cord of a baby, well, then those cells can be used to help that particular baby later on. Okay, next slide. So be aware of this on a test. 2 reasons for cloning: reproductive, therapeutic. Okay, don't care. Okay, Dolly. You should at least know this one. Dolly was the very first sheep that was cloned. So Dolly is actually a cloned sheep. Okay, so be aware of Dolly, the cloned sheep. It's not like the other sheep. It's exactly the same as the other sheep. It's cloned. Okay, this slide tells you all the things we have had the opportunity to clone. So we've cloned mice, horses, dogs, mules, cows, pigs, rabbits, ferrets, camels, goats, and cats. You do not have to memorize this slide, just have a general idea of what kinds of things have already been done. Again, not a biggie. Here's a picture version on page 207. Again, just have an idea, don't really care. Now, why do we do this cloning? We do it because some animals are endangered. And as they are becoming endangered, we want to make sure we don't lose them, so we often clone. So cloning is often to protect the endangered animals. Okay. Alrighty. This is already something we've talked about. Remember I said cloning for endangered animals to restock the population. Alright, and to produce certain traits, so like in agriculture, you want to have certain traits in plants. Okay, same way. Now, this slide's important. We're going to go down this one line by line. It says cloning may also create new problems. Well, here's what often happens. Conservationists say this. They may detract from efforts to preserve their habitat, because now that we know how to make these animals cloned, we worry about just cloning the animal and not worrying about the habitat that they come from. Number 2, it does not increase genetic diversity, because remember, we're making a perfect copy of that previous animal. So there's no genetic diversity in that. It's a perfect copy. And therefore, not as beneficial to endangered species, because as we have endangered species and we make another copy, all we're doing is keeping the same genetics over and over and over, so the genetic pool never changes and each animal is exactly the same as the other. So if there's something bad as a trait in one of the animals, all we're doing is making a perfect copy that's exactly the same badness in each one of them. There is no way to lose some of these traits. So like we saw in the other chapter, there are ways that sometimes it'll skip a generation. Well, not this time. This time, every single animal, since there's a perfect clone of each other, will be exactly the same. So any flaws will be reproduced. Okay, next slide. Okay, don't really care. Okay, don't care. We've already talked about that particular slide. Okay, this one I do care. This is where I mentioned where can I get these special cells. One way to get them is through the embryo. So I mentioned something about the umbilical cord. Well, the embryo is one way of getting stem cells. Now, yes, you can also get stem cells from adults, but stem cells from the embryo are better. Okay? So stem cells tend to be better from the embryo versus the adult. Now the next slide on page 208 will explain why. So this is page 208. If you look at the top part of this picture on page 208, we see the adult stem cell, and then it just says blood cells. Because adult stem cells do not have that much of a capability. Okay? They don't know how to make different forms. When I say they, meaning the cells themselves don't have the roadmap to make other kinds of cells. So it's limited. Whereas the embryo stem cell, that embryo stem cell is very open. It can be turned into a lot of different kinds of cells. So people with brain damage, well, there's a way to help grow nerve cells in the brain. People with leukemia, there's a way to help with leukemia. People with muscular dystrophy, there's a way to help with that. Or people with heart attacks. So if you had multiple heart attacks and your heart is damaged, well, there's a way to help you with that. Now, these cells are called differentiated cells. So embryo stem cells can produce a lot of differentiated cells. Okay, that's an important term for the test. Stem cells don't do so well from the adult. So adult stem cells don't do so well, but the embryo stem cell can make many different differentiated cells. That's important from this slide. Okay, next slide. Okay, another place that you can get blood from is the umbilical cord, which I've already mentioned a couple times. Okay, so you can get blood from the umbilical cord as well. And here is a baby's umbilical cord, and they drew blood from that. Now, that blood can be used as stem cells to help that child in the future. Okay, so it can be used to help that child in the future. I've already mentioned adult stem cells. Okay, now we're on this slide, page 209. Page 209. Now on this slide, we have terms that you need to know. I'm going to start with the second term because that's the sequence it should have been. The first term for us is proto-oncogenes. All of us have some degree of proto-oncogenes. These are the genes that can turn into a cancer later. So all of us have some degree of proto-oncogenes. We either got it from our environment, so if Exxon or refinery or Mobil refinery is your next door neighbor, and you've got all these pollutants and these cancer-causing chemicals out there, well, that's going to be the environmental reason for having proto-oncogenes. The other way you get these proto-oncogenes are from your parents. So your parents will pass on these proto-oncogenes to you. Now, what's the other term on the top there? Oncogene. Well, a proto-oncogene turns into a oncogene. So a proto-oncogene will turn into a oncogene. Now, just because you have an oncogene does not mean you're going to get cancer. It just means you're now very potentially going to get cancer. So proto-oncogenes turn into oncogenes. The oncogenes have to be expressed badly. And we'll see that on the upcoming slide. So there has to be something wrong in order for the oncogene to now make a bad protein. Now, one of the things I've already mentioned to you that has to happen for any form of cancer pretty much is you have to mess up the p53. Remember, p53 was a tumor suppressor gene. Okay, so we're going to see that on the next upcoming slides. Page 209 is coming up next. This is page 209. We're going to go from left to right on this picture. Here we have at the top the proto-oncogene that I mentioned that you inherited from your parents or you got it from the environment. The first one says a mutation within the gene. So somehow you messed up the p53, as an example, p53. The tumor suppressor gene is not working anymore, and now your oncogene is free to make some hyperactive protein, something bad. Okay, something bad. Then over here, here's a normal situation. And then over here is another normal situation. Okay. But notice, we started with a proto-oncogene. It does make an oncogene, and only when that oncogene makes something bad do you end up getting a cancer. Okay. And usually it's because you messed up the p53 that you're now making something bad. Proto-oncogenes themselves do not make cancer. So proto-oncogenes themselves don't make cancer. Even oncogenes by themselves don't make cancer. It's only the bad protein that comes from them that makes the cancer happen. Okay, next slide. Bottom of page 209. Now we're going to talk about the tumor suppressor gene, which is that p53. So p53, tumor suppressor gene, page 209's picture. And that's this one. And as I've already mentioned, as long as the tumor suppressor gene, that night watchman, is doing its job, you're going to be okay. The moment the tumor suppressor gene is sleeping on the job or not doing its job properly, the mutated tumor suppressor gene will now make a bad protein, turn this thing into an oncogene, and now we have cancer. So cancer does not just happen automatically. Okay. All right, next slide. Okay, don't care. All right, now page 210 talks about the multi-step process. So as you can see, the first thing that has to happen is a proto-oncogene. This is page 210. What has to happen is a proto-oncogene on the far left has to turn into a oncogene. Then the oncogene has to now have a messed up tumor suppressor gene. See how it says tumor suppressor gene is inactivated. That starts the first form of cancer. Then if you mess up more and more tumor suppressor genes, then the cancer gets worse. Then it goes from a benign tumor to a malignant tumor, then it spreads to the rest of the body. And this is an example of colon cancer here. Okay? So it takes a multi-step process for that to happen. Okay, next slide. Now this is page 211 at the bottom. And this is letting you know that certain cancers are not just one change. It takes multiple changes in some cases. So here we have a mutation on one gene. Then we have a screwed up mutation on another gene, and then a third, and then a fourth. So different chromosomes, multiple mutations, multiple mess ups of tumor suppressor genes, and then you end up getting a cancer. So a cancer is not an overnight process, and it's not just a one-step process. Many things have to go wrong before the cancer arises. And p53 happens to be the starting point in many. Okay. Don't care. Don't care. Okay, here's Angelina Jolie, and the reason why she's mentioned in our textbook is because of the BRCA gene, which is what I'm going to back up to. And that's this one right here, which I don't really care for, but it's to let you know that you can't inherit the cancers. Remember I mentioned you can either get the oncogene from the environment, like the proto-oncogene from the environment, or you can get the proto-oncogene from your family, which then makes your oncogene chances even higher. So you can inherit a cancer. So cancer can be inherited. Okay, now we go to this slide. Now, in a previous chapter, we mentioned mutations and mutagens. And at that time, in the other chapter, I said mutagens are simply something that causes a mutation, a change. And if that change causes a cancer, then it's called a carcinogen. And that's the same thing mentioned here. So we have issues with cancers, but cancers can only arise if you have mutations. If you never have a permanent change in a gene, and the gene is messed up, and then the gene is fixed automatically, and no one knows anything better, then you're not going to get a cancer. It's only when the mistake is not fixed, and it just goes on cell after cell, mutation after mutation, then you're going to get a cancer. So carcinogens are chemicals that can cause a cancer. Okay, so carcinogens are things that can cause cancers. One of the ones that if you're bored you can look up, you can either look it up on Google or you can look it up at the grocery store, and that is a cereal called Nabisco shredded wheat. It only has 2 ingredients in Nabisco shredded wheat. It's 100% whole wheat, and the other ingredient is BHT. Now, BHT is a chemical. It's a preservative. It's found in the lining of the bag that your cereal is found in. And many cereals do have BHT or some variety of it like BHA or BHQ. And it is known that BHT, which is found in Nabisco shredded wheat, and there's only 2 ingredients, is known to cause cancer for the liver. And countries like the Netherlands, the Dutch, they use that a lot for preserving their cereals and their grains. And it's known to cause liver cancer. So carcinogens can cause cancers, and they include UV radiation and tobacco products. Now this chart here I do not care for. It's on page 212. But what I want you to understand, instead of memorizing this chart in any way, and I don't really care for the chart, but I want you to understand there's 2 categories. The 2 categories are hormones. Okay, so there are cancers caused by hormones, and then there's cancers caused by the environment. Let's go down this list, and I'm just going to show you an illustration of what categories they belong to. You do not need to memorize that for the test, but just be aware there's 2 categories. There's those hormone related, and there's those that are environment related. I'm just going to briefly go down this list. So breast cancer would be hormone related. Prostate cancer, hormone related. Lung cancer would be environment. Colon and rectum would be environment. Skin, environment. Lymphoma, environment. Bladder, environment. Uterus, hormone. Kidney, environment. Leukemias, that would be the environment. Pancreas, environment. Liver, environment. Brain and nerve, that could be both environment and hormone. Stomach, environment. Ovary, hormone. Cervix, hormone. So you see that we have only 2 main categories, either a hormone or an environment-related cancer. Okay, next slide. Don't really care. Don't really care. Here's a picture at the end of the chapter on page 213 showing you a bone cancer there. That's a bone cancer. Don't care. Don't care. Don't care. Don't care for the end of the chapter stuff. And we've already talked about what is pertinent. Okay, this is the last picture of chapter 11's video. And that's page 214. We're going to go around this, and this will be the end of chapter 11's video. So 214. We're going to go around the clock. So we're going to start up here. We have a proto-oncogene. One of the ways, as I mentioned, that a proto-oncogene turns into an oncogene, and then that causes a bad protein, which causes cancer. The other way is a proto-oncogene is normal, everything's fine and dandy, but then the tumor suppressor gene. So the tumor suppressor gene mutates, and that causes cancer. So either you got it because of the oncogene making a bad protein, or you got it because the tumor suppressor gene is messed up, and then that's how you get cancer. All right? So that was page 214, end of chapter 11's video.